Welcome. Let's look at the sperm cell. The sperm cell is also referred to as the spermatozoon. The spermatozoon is the singular of the sperm cell, while the plural is the spermatozoa. It is a microscopic male sex gamete that is produced in the testes. After its production in the testes, it is released via the male ductal system, where it is finally emptied out into the female reproductive system for it to initiate the process of fertilization. The entire length of the sperm cell is about 60 microns. It is produced through a process called spermatogenesis in the testes. And this, of course, is under the influence of hormone, and it tends to undergo structural changes from one form to the other before we finally arrive at the mature sperm cell that is referred to as the spermatozoon. This sperm cell to unite with the female gamete that is known as the oocyte or the ovum to form the zygote. We've done a lecture on the process of fertilization. You can go and check the structural basis behind fertilization up in our previous lecture to upgrade our knowledge. The sperm cell is seen to have 4 regions. The first one is the head region, and this region is the head region of the sperm cell, followed by the neck, which is a constriction that is followed after the head region. Then more distally, we have the middle piece or the mid piece. Then lastly, the terminal portion, we have the tail. So we have 4 sub-regions, and we'll be looking at these regions one after the other to see what their structural basis is and also the functions that they present. Talking about the head of the sperm cell, we said that this is the head of the sperm cell. This is the most superior portion and the rounded part of the sperm cell. And in this head region, we have the nucleus. This is the nucleus, a centrally placed nucleus that is very large. And enveloping this nucleus, we have the nuclear envelope that tends to provide a protection or a covering to the nucleus. Within the nucleus, we have chromosomes. The chromosomes are genetic materials that are responsible for the transmission of characters from parents down to the offspring. And this is 23 in number. So the mature sperm cell, we have 23 number of chromosomes. And at the point of fertilization, it's going to unite with the female egg, which also has 23 number of chromosomes, and that is the haploid number of chromosomes. And this will unite to form a diploid number of chromosomes. So when you have 23 plus 23, you'll be having 46 chromosomes. And this is the normal presentation that is seen in human form. So you have half deposited from the male gametes and also half deposited from the female gametes, both coming together to form the normal 46 chromosome that is needed for developmental process to occur. So we said that the entire length of the mature sperm cell is about 60 microns. And from this, so out of the entire length of the sperm cell, we have 5 microns in length that is being taken up by the head region, which means that the bulk of the length of the sperm cell is seen to be occupied by the middle piece and of course mostly by the tail region. So if you look more in the head region, apart from the nucleus taking a large portion of the space, we have the acrosome that is seen in the anterior 2/3 portion of the head region. This acrosome has 2 layers. We have the inner layer, and this is the inner layer of the acrosome, and we have the outer layer of the acrosome. This layer tends to be continuous so as to create accommodation for the acrosomal enzymes. So they to form like a sac kind of, and within the sac, we have the acrosomal enzymes. We know the function of this acrosome during the process of fertilization. We've discussed this extensively in our lecture on fertilization, where it enhances the passage of the sperm cell through the coronal radiator cell and also allows it to be able to break through the zonal pellucida, which is the shell-like covering of the egg. So this acrosome is very important in the process of fertilization because without it, the sperm will not be able to penetrate into the egg to form the zygote. So going to the neck region, more distal to the head, we have a constriction that is called the neck. This is also referred to as the connecting piece. Connecting piece because it is this region that connects the head with the remaining part of the sperm together. So it is this neck region that tends to create like a connection point between the head region and the extension that comes from it. So this is a very short region of the sperm, and you see it highlighted in red. So we have two structures contained within this region. We have the proximal centriole, and this is the proximal centriole. And we have the distal centriole, and this is the distal centriole. And what they do is that the proximal centriole is responsible for spindle formation. After fertilization, tends to go with the head of the sperm into the cytoplasm of the egg to help in the formation of spindle arrangements. While the distal centriole is important in motor movement, the distal centriole will create like an extension from it that runs through to the terminal part of the tail. So what happens basically is that under ribosomal production of tubular proteins, we already know that the ribosome is responsible for protein synthesis. So it tends to synthesize proteins, and these proteins are tubular proteins, which are deposited at this region, and they tend to be connected from one end to the other. And that is how the extension is brought about. So it tends to like lengthen the extension that is created from the distal centriole. So while the proximal centriole contributes or follow up with the head into the cytoplasm of the egg by helping to initiate further progression of developmental process by the formation of zygote. This is solely responsible for movement because it creates an extension from it that runs through the entire length of the sperm. And this is what flips and helps to propel the sperm. The next region is the middle piece. After the constricted neck region, we have the middle piece region. And the structural content of this region are filaments, and this is highlighted in black. And you see it running through the middle piece. Another important structure that is seen within the middle piece region is the spiral arrangement of mitochondria, and this is highlighted in red. So you see a central core filamentous structure. You see a spiral arrangement of mitochondria around it, and this is what is presented in the middle piece of the sperm cell. The mitochondria, what it does is to produce energy for the sperm to be able to flip so as to push the sperm through the path they move, because we know that motility is important during the process of fertilization, because where they are produced is far distant from where fertilization will take place. So they need to run through a path, and this can only be done if movement is assisted or supported. And this will be supported through the ATP that is produced by the mitochondria. So you see a number of mitochondria in this region that helps to produce ATP that the tail can use up to generate energy so that they will be able to give a propelling action for this sperm cell to be able to move through. Then more distally, we have the tail. The tail is the most terminal portion of the sperm cell, and this is the tail region. From this region, after the middle piece region, down to the terminal point of the tail. So you have the tail, and the tail, of course, takes about 50 microns from the 60 microns in length of the entire sperm cell. You can see that the bulk of the length of the sperm cell is taken up by the tail, and we can justify this because of the action that they need to exhibit in terms of movement. So this tail is like a modified or a specialized type of cilia called the flagella. It is long and it is slender. So what it does is to provide a wave-like pattern of movement so that they will be able to help propel the sperm. Also, for us to know that the sperm cell is the only cell in the human body that has flagellum. If we look more deeper, let's say we cut a T-section around this part. This is the kind of presentation we would see. This tail is made up of collection of microfilaments. And the microfilaments, of course, are arranged in specific patterns. You have filamentous arrangements of microtubules arranged in 9+2 pattern of arrangement. So this is what you see. You have two centrally placed microtubules, and this is highlighted in blue, or we can say that we have a pair of microtubules. So they are located at the center, while surrounding it at the peripheral, we have the peripheral arrangement that is seen in 9 pairs. So you have 9 pairs of microtubules arranged around the central tubules, and this arrangement is called the peripheral tubules. So this is the pattern by which they are arranged. So when you take a T-section of the extension of the sperm cell, this is the kind of presentation that I would see. And this arrangement is called the axoneme. So furthermore, on the tail region, the tail we said can be further subdivided into two sub-regions. We have the principal piece, and this region is the principal piece, and we have the end piece, and this is the end piece, the most terminal part. So the initial region of the tail region is called the principal piece, while the terminal end is called the end piece. There is a form of difference between the structural components of these two regions, and it is on this basis that they are being subdivided. We already said that we have an extension from the distal microtubule that is located in the neck of the sperm cell. This is the constricted neck region. So you have an extension extending from this region, and it passes through the entire length of the extension from the head. So these tubules at the middle piece, we already know that it is surrounded by a spiral arrangement of mitochondria. That is what you see in the middle piece region. So as this extension goes further, when it gets to the principal piece region, it is surrounded by a fibrous sheet. And this is what is highlighted in green color. And this is what is expressed in this image down here, where you have the 9+2 pattern of arrangement enclosed by fibrous sheets. So this is the fibrous sheet highlighted in green surrounding a microfilament arrangement. You see that the extension deep of microfilament is highlighted in black. While covering the black extension, we have the fibrous sheet that is highlighted in green. And that is the kind of presentation that we see in the principal piece of the tail. When you go more distal, you also have a filamentous extension to the end piece, but this extension is not covered by fibrous sheets, and that is why the green coloration is not seen at the end piece region. The green coloration represents the fibrous sheet, so it does not extend to the terminal part of the tail, which is called the end piece. So the difference between the initial segment, which is called the principal piece, and the terminal segment of the tail, called the end piece, is the loss of fibrous sheet. So what is seen in this terminal region is just the arrangement of filamentous microtubules that is covered by the plasma membrane. We know that the plasma membrane encloses the entire structure of the sperm cell. Then clinical application, we have to talk about the sperm morphology. Sperm morphology is one of the basics in establishing sperm parameters. When you want to go for fertility assessment in male and they need to check sperm parameters, one of the properties that will be evaluated is sperm morphology. And since this lecture is about the anatomy of the sperm, it's good for us to relate what the morphology is in evaluating fertility assessment. So sperm morphology basically describes what the sperm looks like. We already know the basics. They need to have a head, a structured neck, the middle piece, and also the tail. So these are the 4 basic structures that the sperm needs to have. And of course, within those structures, we also have specific elements that are contained within them. And the sperm also should present that. But when you now begin to see sperm that lack 1 or 2 of these regions, maybe you have a sperm that does not have a neck, or you have the one that has a neck but does not have a tail, or you have a sperm with 2 eggs, or you have a sperm that is seen to be very small in terms of size, or you have a sperm cell that has 2 tails. So this kind of morphological inactive, so they will be unable to function as they should. This will tend to reduce the fertility of the person. So thanks for watching this video. Let's meet in our next lecture class.